We're going to discuss a paper published in the British Journal of Cancer in 2004, which is basically addressing the use of DNA vaccines, naked DNA vaccines, in treatment of prostate cancer. Now, the purpose of the study was to determine the safety and immunogenicity of a naked PVAX-based DNA vaccine containing the tumor antigen PSA. Now, how this vaccine was constructed, it was constructed using the full-length human PSA gene inserted into the PVAX vector commercially available from in vitro gen. And sequence analysis was performed to ensure that the insert put into the vector was the correct PSA insert. The vaccine was produced under GMP with less than 10 European units per milligram of endotoxin, more than 85% supercoiled DNA, protein content less than 10 micrograms per milliliter per milligram, and chromosomal DNA content less than 30 micrograms per milligram of plasmid. The vaccine was produced and stored under the guidelines from the NIH involving recombinant DNA molecules, and it was stored frozen at -80 until use. The potency of the vaccine after it was made was assessed by in vitro transfection into co cells and intramuscular injection into mice, just to make sure that the plasmid actually induces the protein, the expression of the protein. Now, the clinical trial. 9 patients with advanced prostate cancer were recruited. These patients, to enter the trial, they had to have progressive disease and to be resistant to known therapies, including androgen ablation. The patients had to have a ECOG performance of at least 2 and a life expectancy of at least 6 months. How the patients were treated. 3 patients were randomized per group, 3 groups. Each group was given an escalating dose of DNA vaccine, of the PVAX with the PSMA inside of it. The total concentration was 100 micrograms of plasmid, 90 micrograms given intramuscularly, and 10 micrograms given intradermally in Group 1. Group 2 had triple the concentration, and Group 3 had another tripling of the concentration. The vaccine was administered into the deltoid region, and it was administered 5 times at 4-week intervals. Now, together with a vaccine, there was given an adjuvant. The adjuvant used was GM-CSF and interleukin-2, a recombinant, which are both clinically available. GM-CSF was given at 40 micrograms per day, starting at day -2 and ending at the day of injection. And interleukin-2 was given subcutaneously, starting the day after the injection, the day after the vaccination. In the same extremity as in the extremity where the vaccine was given, for 7 days, starting the day after vaccination, for 75 micrograms per day. So if you take a look at the schema, at day -2, day -1, and day 0, GM-CSF was given 40 micrograms per dose. At day 0, the vaccine was given intramuscularly and intradermally. And the day after the vaccine, interleukin-2 was given subcutaneously for 7 days. And this cycle was repeated a total of 5 times, 4 weeks apart. Immune response was assessed before and also assessed at week 5 and after the immunization cycles have been completed. When the peripheral blood lymphocytes of the patients were taken and stimulated outside of the body with PSA, interferon gamma production was seen only in C, only in the patients having received the highest concentrations of the PVAX PSA DNA vaccine, as you can see where it says C. The other patients appeared not to have induced an interferon gamma response. Antibody production was seen at all levels of DNA vaccination. However, the issue here is that you see it before and after the antibodies produced. So because of this, your vaccine isn't really inducing production of antibodies, except in patients 7 and 8, where you can see increased antibody response after the vaccination. And most importantly, the impact on PSA. Here you can see A is the patients receiving 300 micrograms, B is the patients receiving 600, and C is the patients receiving 900. And as you can see, it appears that in patient number 9, there was a reduction in the PSA. In the other patients, the vaccination maybe made a response, maybe didn't. Now, in conclusion, safety was demonstrated in this study. There was an idea that immune response can be generated, but more experiments are needed to draw clinical conclusions.